Welcome to the second screencast on enzymes. This looks at inhibitors and cofactors and coenzymes, as well as the effect of inhibitors as poisons and also as drugs. So, dealing with inhibitors first of all, there are various ways that we can classify inhibitors. We can have competitive inhibitors and non-competitive inhibitors, and we'll look at this first of all. So overall, an inhibitor is a chemical, a molecule which somehow reduces the rate of an enzyme-controlled reaction. Okay, and there are two principal types: competitive and non-competitive. We're going to deal with competitive inhibitors in this slide. A competitive inhibitor is similar in shape to the substrate, and therefore it can also bind to the active site of the enzyme, forming what we call an enzyme inhibitor complex. The effect of these is that therefore less enzyme substrate complexes can form, because some of the active sites have formed enzyme inhibitor complex, and therefore that results in less product. Okay. However, if we keep on increasing the substrate concentration, what happens is that there becomes less and less probability of an inhibitor binding to the active site to form an enzyme inhibitor complex, and therefore eventually you can overcome that inhibition, and I'll try and illustrate that below. First of all, though, I've got a little animation which shows a competitive inhibitor binding to an active site.
Competitive inhibition occurs when an enzyme encounters a blocker which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur.
Okay, now if you can imagine this scenario here, where I've got an enzyme here, I've got a substrate molecule here, and I've got an inhibitor molecule here. So first of all, the idea is that here is the substrate, and that can bind into that active site there. Okay. However, our competitive inhibitor can also bind into this active site here to form an enzyme inhibitor complex, and in so doing, the substrate can no longer bind to that active site. Okay. Now, in a solution, these enzymes and these substrates and these inhibitors will all be moving around with a certain amount of kinetic energy, and in this case there is equal likelihood of this binding to that as there is this binding to that. Okay. So obviously if you multiply this, if there were lots of enzymes in this solution and a number of inhibitor molecules and a number of substrate molecules, okay, only half the number of enzyme substrate complexes would form, because half of the active sites at any one time are as likely to be occupied with inhibitor as they are with substrate. Okay, so we would reduce the rate of this enzyme controlled reaction. Okay, however, the effect of the inhibitor becomes less as you add more substrate in. Okay, so in this case, the inhibitor only has a 1 in 3 chance of colliding and binding with that enzyme molecule. Add some more, it's only got 1 in 4 chance. Okay, and so on, and we keep adding more and more and more substrate, and we get to the stage when there is so much substrate that the inhibitor actually has very little probability of binding to the active site as compared to the substrate molecules. Okay, so by adding more and more substrate, you can overcome the inhibition by this inhibitor. Okay, with this competitive inhibitor. Okay, and if you represent that graphically, here I've got my normal curve for my enzyme with substrate with no inhibitor present. Okay, now here I've got another curve. The green curve shows the effect with the substrate with a competitive inhibitor added. Okay, so we've got our substrate concentration increasing, and at low substrate concentrations there is a degree of inhibition. Okay, because at these low substrate concentrations the inhibitor has quite a high probability of colliding with the enzyme and making enzyme inhibitor complexes. As the concentration increases, however, there are so many substrate molecules there that the inhibitor has progressively less and less probability of colliding with and binding with an enzyme to form an enzyme inhibitor complex. Okay, so we can overcome the inhibition of a competitive inhibitor by adding more and more substrate. Okay. Now the other thing that this slide requires you to know is the difference between reversible and irreversible inhibitors. Okay, if I go back to the top here, it says with reference to both reversible and non-reversible inhibitors. Okay, this basically means that I can illustrate this below. Okay, if this was some inhibitors are what we call reversible inhibitors, and what they do is that they come and bind to the active site, okay, and they form weak bonds, and there's also a fairly temporary association, so it might come in, bind, and then diffuse away again, and come in again, bind, diffuse, come away again, because the bonds are weak, and they're also temporary bonds that occur between the inhibitor and the enzyme. Okay, as opposed to a non-reversible inhibitor, what it does is it comes in and binds to this, okay, and forms very strong bonds, often covalent bonds between the inhibitor and the enzyme. Okay, and that's permanent. Okay, so that stays like that. So that's a non-reversible inhibitor. Okay, so hopefully that distinguishes between reversible and non-reversible inhibitors. Okay. So moving to the next slide. This slide deals with non-competitive inhibitors. Okay, so a non-competitive inhibitor binds to a region of the enzyme other than the active site. Okay, so in this diagram here, this is a non-competitive inhibitor binding to this enzyme here, but not in the active site region. Okay, the result of that is it alters the shape. So the active site here is normally looks like that. Okay, and the binding of the non-competitive inhibitor to that has resulted in a change in shape. Okay, and as a result, the substrate can't bind to the active site. Okay, and unlike competitive inhibitors, we can't overcome this by adding more substrate, and I'll explain that in a minute. Okay, if we look below here, this little animation illustrates a non-competitive inhibitor.
Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as a non-competitive blocker is bound to the enzyme.
And as we can say, you can get reversible ones of these, which bind with weak bonds, temporary bonds to here, or you can get irreversible ones which bind with covalent bonds and completely alter the shape irreversibly of our active site. Okay, so the most common form of inhibitor forms temporary bonds with enzyme and are termed reversible. Okay, some inhibitors bind strongly to enzyme and are called irreversible. Okay, dealing with a non-competitive aspect of what they do. Okay, if we look at this illustration here, If I just rearrange these, so here I have an enzyme. Actually, it's the wrong way around. Here I have an enzyme here, okay, with its active site, okay, which can bind, normally it can bind to a substrate molecule, okay. However, if we have an inhibitor molecule that binds to this like that, okay, what it does is it changes the shape of the active site, which means that our substrate can't fit in there, okay. Now, if you can scale this up and imagine what might be happening in a cell or in a beaker or a test tube, we would have this scenario. If I put both types of enzyme in here, one which is bound to an inhibitor and one which is bound to which isn't bound to an inhibitor, and then what I do is if I put a substrate molecule in there, okay, you'll notice that, okay, the enzyme could attach to that but couldn't attach to that, okay. So the fact that this enzyme here is attached to an inhibitor reduces the amount of active sites available for the substrate to bind to, and therefore reduces the chances of an enzyme substrate complex forming, okay, and therefore an enzyme product complex forming, and therefore product being formed, okay. So we reduce our concentration of active sites, okay. If we continue to add, okay, we would increase the chance of enzyme substrate complexes forming with this enzyme here, okay, but it would have no impact on the chances of an enzyme substrate complex forming with this enzyme here, okay. So increasing the substrate concentration in this case, okay, will initially increase the rate a little bit, okay, but it will never get a maximum rate of enzyme controlled reaction or the same rate as we would without the inhibitor, because at any one point some of the enzymes will be bound to inhibitor molecules, and therefore the active sites will not be able to form enzyme substrate complexes, okay. If I represented that graphically, okay, so here's my normal line of an enzyme controlled reaction with a substrate only, but this time I put a non-competitive inhibitor in, okay. So at low concentrations we increase the rate, okay, because there's more substrate, there's more chances of collisions with the active sites that are available, okay, but the maximum here is never reached, okay, because at any one point a certain proportion of the active sites will be unable to bind to substrate, okay, because the non-competitive inhibitor has bound to its sites and therefore have rendered the active site unable to bind to the substrate, okay. So in summary, okay, so these non-competitive inhibitors, they bind to a region other than the active site, alter the shape of the active site, okay, means that enzyme substrate complex can't form, and therefore no product formed, okay, and it cannot be overcome by increasing substrate concentration, okay. So that's the business about non-competitive inhibitors, and here's a bit about reversible and irreversible, okay, and inhibitors, most inhibitors are reversible, okay, temporary bonds, weak temporary bonds with the enzyme, whereas some inhibitors attach very strongly to the enzyme and form permanent bonds, and we call those sort of irreversible inhibitors or non-reversible inhibitors, okay. Moving on to the next slide. Okay, this slide looks at the idea of cofactors and coenzymes, okay, and also we mentioned prosthetic groups here as well, which you can also, we've come across these in in hemoglobin, but we can also get prosthetic groups in enzymes. Okay, so so dealing with coenzymes and cofactors. Okay, many enzymes only work if there are some non-protein cofactor involved. Okay, so it's a bit of the enzymes are predominantly proteins. Okay, but sometimes they have to have non-protein chemicals or molecules that bind to the enzyme for the enzyme to work. Okay, so sometimes these are cofactors, which are inorganic molecules. Okay, so chloride ions, for example, are required to bind to saliva amylase for it to work to break down starch. Okay, and there are other small cofactors which are inorganic ions. Okay, and then also, so if we look here, we've got cofactors which can be broken down to either inorganic or metal ions. They don't have to be metal ions, they can be inorganic ions. Okay, and what they do is that they either bridge enzyme to the substrate, okay, so they sort of attach somehow the enzyme to the substrate, okay, or they combine with the protein and act as a catalyst within that protein. Okay, whereas coenzymes tend to be larger organic molecules, okay, and they tend to bind alongside the substrate to the active site. Okay, so for example, down here, here's an example of what a coenzyme might do. Okay, here's an inactive form of an enzyme. Okay, but the coenzyme has to bind to the enzyme in order to be able to make the shape of the active site for the substrate to fit into. Okay, so without this coenzyme, the substrate wouldn't fit into this active site. Okay, so that's coenzyme, which tends to be a large organic non-proteinous molecule. Okay, so that's what a coenzyme does. As well as that, you can have things called prosthetic groups. Okay, these are things which are permanently attached to the protein molecule, the enzyme. Okay, and they're important in the catalytic function of the enzyme. Okay, so the prosthetic group we've come across before is the heme group in hemoglobin. Hemoglobin is obviously not an enzyme though. Okay, but prosthetic groups can also be bound to large protein enzymes. Okay, so the example I've got below here is here I have a molecule of carbonic anhydrase, which you've come across in unit F211. It's the enzyme that we find in red blood cells, which converts carbon dioxide into carbonic acid. Okay, and it's a large protein, globular protein. Okay, lots and lots of tertiary folding. Okay, but its active site in here has got a zinc ion. Okay, which is attached by these three side groups of a particular amino acid called histidine. Okay, it's kept in place there, and this is the bit which does the catalytic center. Okay, so carbon dioxide and water bind to this center here to be converted into carbonic acid. Okay, so that's the prosthetic group in carbonic anhydrase. Okay, an enzyme you've come across before. Okay, so those hopefully distinguishes the various cofactors and coenzymes. Okay, a cofactor is a non-protein factor. Sometimes these are small ions. Okay, we call those cofactors. Sometimes they're larger organic molecules. Well, they're still quite small organic molecules, but obviously larger than metal or non-metal ions. Okay, which bind temporally to the active site at the same time as the substrate. Okay. Sometimes they're involved in carrying chemicals from one enzyme to another. Okay, enzymes often work in what we call metabolic pathways, and what happens is that one enzyme converts a substrate to the product, and then that product is carried to the next enzyme where it becomes a substrate for the next enzyme, and it's carried there by a coenzyme. And you'll come across that when you study respiration in the upper sixths. And lastly, prosthetic groups, which are permanently attached group to a protein. Okay, in this case we're interested in enzymes, and the example of we've got a zinc which is attached to a zinc ion which is attached to a carbonic anhydrase to form the catalytic center of the active site for the enzyme which catalyzes carbon dioxide and water to carbonic acid. Okay, so moving on to the next. Okay, it says state that metabolic poisons may be enzyme inhibitors and describe the action of one named poison. Okay, this is just something you've got to learn. Okay, I'll go for the first one here. So the cyanide is an irreversible inhibitor. It's a irreversible non-competitive inhibitor, okay, which binds to the enzyme called cytochrome oxidase, which you've not come across before, but it's one of the key enzymes involved in respiration. Okay, so cyanide binds to cytochrome oxidase. Okay, respiration stops. Okay, and therefore it's obviously a metabolic poison, because respiration obviously is an important metabolic process. Okay, so cyanide and cytochrome oxidase. I'm afraid these things you've just got to learn. It's an irreversible, non-competitive inhibitor. Okay, you just have to learn that, and I would go for that first one. Okay, because you could be asked for that in an exam. Moving to the next one. Okay, this one you've got to state that some medicinal drugs work by inhibiting the activity of enzymes. Okay, there's lots of drugs that work in this way, and some blood pressure controlling drugs called ACE inhibitors, angiotensin converting enzyme inhibitors are inhibitors. We've got some neuroactive drugs, monoamine oxidase, MAO inhibitors. Okay, but anyway, the one I've hit upon here, which is mentioned in all the textbooks, is penicillin, which is a drug you've all heard of before. It is a drug which is involved in fighting bacterial infection, and its site of action is an enzyme called transpeptidase. Okay, now bacteria have a cell wall. Okay, and the cell walls are made of a substance called murin. Okay, and murin is essentially polysaccharide chains, okay, which are held together by short polypeptide chains. Okay, and the enzyme that makes these polypeptide chains, or links these polypeptide chains to the strands of polysaccharide, is called transpeptidase. Okay, and the penicillin molecule and lots of antibiotic molecules are inhibitors to the transpeptidase enzyme, which means that bacteria can't make cell walls. Okay, without those cell walls, what happens is that they burst due to osmosis. Okay, so the water rushes into the cell along a water potential gradient, and the cell bursts because it hasn't got a cell wall to withstand that pressure. Okay, a little animation below to illustrate that.
Ring. This structure is capable of binding to the enzymes that cross-link peptidoglycans. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial cell wall synthesis. By inhibiting cell wall synthesis, the bacterial cell is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid cell wall, these cells burst when subjected to the low osmotic pressure of their surrounding environment. As well, the antibiotic penicillin binding protein complex
So the details of the structure of a murine cell wall are not important here. You just need to remember the word transpeptidase and the fact that penicillin is an inhibitor of transpeptidase, and that disrupts cell wall production in bacteria, which cause them to burst, which is the way that penicillin antibiotics can kill bacterial cells. Okay, so that ends our second screencast on enzymes.